Hi, здравствуйте.
Здравствуйте. We are really excited to hear your talk on clinical genome analysis platform.
Thank you. Thank you, Daria, for the introduction and for inviting me here. So today I will tell you about the clinical genome analysis platform that we are developing in collaboration between Brigham and Women's Hospital and Harvard Medical School to enable genome research and precision medicine. So as you probably know, there are two main arms of human Mendelian genetics. There is the clinical genetic diagnosis, where we have a patient that comes to the clinic with known genetic disease. Like in this example, we have a patient with familial breast and ovarian cancer. This patient is being tested by genome sequencing, looking at known genes that are associated with the disease. And there is a mutation that could be found in one of the known genes like BRCA1 or BRCA2. There is also the gene discovery research, where researchers are looking at cohorts of patients and controls with disease of unknown genetic etiology. And by looking at these cohorts, researchers could do new genotype discoveries, phenotype expansions, and provide new molecular diagnostics to those diseases. What we're trying to do with our platform development of the clinical genome analysis platform is to connect these two arms and to enable actual collaboration between the gene discovery world and the clinical genetic diagnostics world. And we are doing it by providing a platform that can support both arms and enable collaboration. So we started 2 years ago in June 2019, and this is basically a genomic medicine initiative that was founded by the Department of Biomedical Informatics, and you saw several colleagues like Paul Aviak from the department, with close collaboration with Brigham Genomic Medicine at Brigham and Women's Hospital, which is a Harvard-affiliated hospital just across the street from our department. This is a complete end-to-end solution for research and clinical phenotype collection, annotation of the variants, visualization of the data, and reporting of the genomic findings. It's open source, so completely open to the community. It's a semi-automated AWS-based bioinformatics pipeline with ingestion to a web application that facing to the users that can do semi-automated interpretation for variant reporting. This workflow was previously used for analysis of cases from NIH-funded projects like the Undiagnosed Disease Network and the Phase-Based Consortium. And this workflow resulted in the past with multiple diagnoses and more than 35 gene discoveries and phenotype expansions. We are using an existing AWS infrastructure, which was under the MIT open source license, that was developed by the 4D Nucleome Data Coordination and Integration Center, led by Professor Peter Park. And we are currently expanding our product portfolio for somatic tumor profiling. And also there is a separate initiative that we are working very close with for single-cell RNA sequencing analysis. And the PI there is Peter Harchenko from the department. So what I would like to do is to provide you with an overview of our workflow and then walk through an example that can give you a feeling of how it feels to actually use this platform. So the workflow starts by having a sample from a patient with genetic disease, either for clinical reporting or for research purposes. The sequencing, the whole genome or whole exome sequencing is done elsewhere. We are not providing the sequencing result services, but the sequence data is being uploaded to our platform and then analyzed with our bioinformatics pipeline and ingested to our web interface platform, where the user can use it for interpretation and generating reports. We are a completely web-based application, so the user doesn't need to install anything on their computer or run any software on their computer. They are getting a secured login, and they are using our platform completely web-based. So you can see here several screenshots, but I will also go with you through an example. And in this example, I would like to show you a case which was referred for clinical interpretation, but similar workflow could be done for gene discovery as well. But what we see here is actually the interactive pedigree viewer. So we can see here the family history of the patient. The patient is 37-year-old male with breast cancer. And we can see here, based on the pedigree, that his sister also had breast carcinoma. His mother had pancreatic adenocarcinoma and breast cancer. His maternal aunt has ovarian neoplasm, and maternal grandfather had prostate cancer. So this is a classical family history of familial cancer. And before going into research, the case was referred for clinical diagnostics. So what I would show you here is a use case for how you can use the platform for the clinical diagnostics. Only the proband was sequenced, since the goal was to look at known genes associated with familial cancer. This is the case overview. So that's how the platform looks like. What we can see here is the pedigree. If we click, we can expand this view. We can see the family information, including the human phenotype ontology, HPO terms that are used to recapitulate the phenotype. We can see the information about the patient, and we can see the workflow. So we start from accessioning to get all the information about the status overview, and then accessioning and report history. Then there is a step of bioinformatics, which is triggered automatically. There is a step of filtering that we will look at right now, step of interpretation, case review, and generation of a report. So right now we are in the accessioning step. We can see here the accessioning of the proband, and if we click on the 12 additional individuals, we can see their accessioning information. And next, I would like to actually click on the bioinformatics to go to the bioinformatics tab and to review how the bioinformatics went. So what we can see here, that based on the quality control metrics, all the metrics passed our quality control threshold. And we can see here the total number of reads, the coverage, the total number of variants that were ingested to the portal. And we can also click at each of the individual steps here to get more information about each of the bioinformatics steps. We can look at the provenance graph. When we click on this, we will get an expanded view of the provenance that shows what are the steps and the intermediate files, starting from the row sequencing FASTQ files here on the left, ending with the annotated VCF here on the right. And I just want to mention that this bioinformatics workflow was previously published. We are using our in-house callers, such as NovoCaller, which are sensitive and specific for the problems at hand. So if we have the Novo inheritance mode, that's what we will see here. And we are using an AWS management tool for bioinformatics workflows, which is open source and developed by our bioinformatics scientist, Sue Lee. It's called Tibana, and it's also available in an open GitHub repository. So when we see that the QC metrics look good, the provenance graphs looks good, we can start analyzing the case. Now we will move to the filtering tab. And at the beginning, there are 3,000 variants, a little bit more, that were ingested to the portal. Those are the variants that fit our hard filter. And those are variants which are coding and variants that were rescued by being pathogenic or likely pathogenic, or variants of uncertain significance in ClinVar, and also variants of potential splicing significance that are based on relatively high splice AI score. So we are basically looking at coding variants plus the whitelisted variants of potential significance for known coding level. Still, it's less than the initial VCF file that has about 3 million variants for one individual, but still too many variants to look at manually. So now we are enabling a filtering process, and what we can do is create our custom filter sets based on the filtering properties here on the left, based on a gene list, variant quality, constraint score, consequence, impact, etc. Or we can load any of the available presets. And in clinical workflow, usually you would like to have a preset which is defined for all case analysis to make sure that analysis is not varied between one case to another and one analyst to another. You would like to actually use something which was predefined. So we can now choose our preset for cancer. And what we will have is this filter block where we can look at genes from familial cancer gene list, which is here. Those are 148 genes, and we can get more information from the tooltip. We would like to look at moderate and high impact, basically looking at missense, splice site, stop gain, stop loss variants. We are not looking here necessarily at 3', 5' UTR and synonymous variants unless they are impacted by splicing. We are looking at relatively high genotype quality and allele frequency, which is higher than 0.5% in the, or in this case, 0.1% in the healthy population. And now we have 10 variants left. So it's good to move from 3,000 to 10 variants, but now we need to go actually and look at the annotations for these variants one by one. So I would focus on one example just to save time. And this is the BRCA2 example. So you can see here that BRCA2 is one of the variants. It's a known breast cancer associated gene. So let's make sure that the quality looks good. So here we have an IGV snapshot that captures the quality. The variant looks good. And then I would like to move to the annotation and interpretation step to actually make notes about this variant. So what we see here is that we can make notes for the variant, for the gene, and make interpretations. And this is the annotation space that provides us with score about allele frequency of the variant, about ClinVar submissions, about predictor tools with potential predictors for the variant being pathogenic, and some external resources. And now I can start and select my clinical interpretation based on the American College of Molecular Genetics guidelines, so ACMG guidelines. So I will choose the rule for pathogenic very strong, for pathogenic moderate, and pathogenic probable. And when I choose those rules, and here are the notes that were made why those rules were chosen, basically pathogenic very strong because of evidence of a splicing mutation within a known gene of high significance, pathogenic pathogenic moderate because the variant has not been reported in NOMAD, and pathogenic probable because in the ClinVar database, this variant has been reported as pathogenic. After invoking these three rules, I have an automated ACMG classification for the variant to be pathogenic. And now I can make a conclusion and send this conclusion to my report. I can do similar things for the genes by looking now at the gene over here and making sure that all the information about the gene meets my hypothesis. This is mostly relevant for research. In this case, we knew that this gene is important. For research, we might want to look at the constraint scores, external databases, and all the additional information that is brought to us in this interface. We would like to make a note, and we would like to send this note to our report and now our internal knowledge base. We also would like to make sure that the variant quality makes sense, and we already did it before starting interpretation, but here is a little bit more information about the variant quality, the coverage, the inheritance mode, and here we have the IGV snapshot if we would like to look at that. So we can make a note now that the variant has good genotype quality, and that provides us with more evidence for causality. And lastly, we can do data exploration for our annotation browser. And the annotation browser is the tool that's used for visualizing these data, not only at the context of the mutation at hand, but also in the context of surrounding area around this mutation. So we can see that this is our mutation. This is a splice site. And we can see that there are several pathogenic or likely pathogenic mutations in ClinVar that have been submitted for other cases. We can also see that there are several pathogenic mutations at the edge of the exon, and we can see that there are several pathogenic mutations within the exon. However, when we go away from the splicing boundaries, there are many benign and variants of uncertain significance, and that makes a lot of sense because that's an intron and non-coding region. And this browser provides us with additional option for making sure that this variant indeed could be classified as pathogenic. For discovery and research, this browser is extremely important because it gives us more evidence where we don't know whether the gene or the variant might be associated with the disease. So looking at ClinVar and also looking at NOMAD in a more visual way, as we do here, helps the researcher or the analyst to make the right conclusion about the case. We use our visualization also for looking at the BOM file browser. So we can now zoom in and zoom out. So this is a BOM file browser with a similar functionality to IGV. And just as Paul mentioned before, in this case as well, we don't need to install IGV or install any tool. We can just easily browse through the genome by using this visualization. And this visualization is very handy for structural variants. So now we already made the decision that the BRCA2 variant is pathogenic, but now our job is to go through the other variants, and we have SNVs, indels, and structural variants. So I will just give you an example for how structural variant could be analyzed using this browser. And in this example, we are looking at a deletion, a heterozygous deletion at mute H gene, which is of importance. And we can see here that there is a 4 kilobase deletion with clear visual impact. So we can see that actually the coverage drops. So when we look at the coverage band, we can see that from high coverage, it drops and then goes up again. And we can also see the soft clips over here, the soft clip threads, that also provides evidence that that's a technically valid deletion. Now we need to do a similar job for the remaining variants. So we looked at BRCA2. That's just one example of additional variant. And now we need to go through all the remaining variants and then go to a case review in order to generate a report. So here we can see in the case review all the variants we went through. Here is our BRCA2 variant. So that's the notes that we wanted to send to the report, the notes about the variant, the notes about the gene, and about the ACMG interpretation. You can see that offline, all the other variants have been also classified. Here is a likely benign, another likely benign. At the case review stage, we can make final decisions about what goes to the report, what is saved to the project, and what would be saved to this case only. And then we are generating our final report. And here is an example for such report in this case, where we can add the partner logo. So whoever is using this platform can customize their own logo. They can customize what we are adding in the header, at the footer, and also the names of the tables. In this case, we are looking at primary findings, actionable secondary findings, carrier risk, pharmacogenomics, and polygenic risk score. And our patient in this case had positive findings for the BRCA2 variant, which is pathogenic, another variant of uncertain significance at the gene pol E. And the subsequent pages of the report have all the additional notes and information about each of the findings and the methodology that we are using. So that's what I have in terms of the platform. That's a very high level of what the platform can do. And again, it could be used for research and clinical purposes. And just to give you a flavor of volume at this point, I just want to say that we are very young in our development. So the minimal viable product was released in January. So we have less than 1 year of experience of production at low scale. But the pipeline, the bioinformatics pipeline, was already successfully used at the Undiagnosed Disease Network to call about 3,000 samples of whole genome sequencing. It was used in a lower scale in the People-Powered Medicine, which is another project of hundreds of individuals right now with whole genome sequencing. It's currently used constantly at the Brigham Genomic Medicine, our co-developers at the Brigham and Women's Hospital. And since July, we expanded the production to include UDN cases from Harvard's clinical site. We have a pilot with Dana-Farber Cancer Institute, and we received the funding from Multiple System Atrophy Coalition to help with analysis, visualization, and interpretation of data for about 3,000 MSA patients around the world. We plan to include many more affiliated hospitals to Harvard Medical School by July next year. We would like to make it completely open and available for everyone in the HMS community that would like to use it. And we are looking at opportunities to onboard 2 or 5 individual clinical labs that will use it in production. For July 2023, we would like to make it open to the community and self-service deployment. It's already open source, but right now it's centralized and required experts to actually run it. It runs at AWS, and it's probably going to be quite hard if some of you will try to deploy it on premises, on AWS account controlled by you. But now what we are working on is truly scaling the system, enabling documentation, making sure that there is automated orchestration via CloudFormation that will help everyone to deploy the system in-house and being able to use it without the need of hiring a team of bioinformatics scientists, data wranglers, and software engineers. And I would like to use this opportunity to thank this team. It takes a village to develop a tool that would be useful. So we have a multidisciplinary team of scientists, engineers, and data wranglers that are developing the system. We are working with our good colleagues at the Brigham and Women's Hospital, and we have a good team of principals that are providing us with strategic direction. And I would like to also thank you for listening, and happy to take these 5 minutes to address some of your questions.
Yeah, thanks, Dana. We have some questions in the chat. one moment. Dana, thank you for the talk. Who can use the data and who has an access to the data?
That's unfortunately highly regulated. So the individual data is being used under the agreements that we have for data usage agreements and IRB protocol, institutional review boards. So the idea right now is that every institution that we are working with will submit a data usage agreement where we define who are the people at Harvard Medical School who can access the data and who are the people in the collaborating institution that are accessing the data. So that would be the data that is including the phenotypes, the medical family history, and the full VCF files. So that's something that is project-based, and we have permissions which are project-based. However, our future goal, one of the features, is to enable data sharing on variant level. So we would like to enable the community to do variant level interpretation, such as people from all projects, whenever they see a variant that someone else, let's say in Dana-Farber, saw before, they would be able to see all the notes and classifications that were made for this variant without the ability to go to the individual sample. So we will not know what is the exact medical and family history of that patient, but we would know that this variant have been seen before in Dana-Farber and Brigham Genomic Medicine. And here is how it was classified, with an option to send an email to this collaborator to actually brainstorm on potential different interpretations or potential discoveries that could be made. But that's a big issue because in the last 10 years, the field of data sharing becomes more and more challenging and more regulated. And you can see it also in the GDPR at Europe and many things that happened in the US with Facebook. They create a lot of limitations on what we can share and how.
Do you plan to distribute the platform, the plain platform, without data? I think
Yes.
The platform provides quite a lot of useful tools that might be useful even without the data.
Yes, Daria. So that's the plan. The platform is already available. So it's already accessible to everyone in terms of the code base. It's available in GitHub. There is a CGAP GitHub repository, and everyone can try and deploy it on their AWS infrastructure. But again, as I mentioned, right now it might be a bit challenging because we were not planning for scale distribution, and therefore the documentation might require a lot of input from the developers. But our goal, it's actually corporate goal to 2023, July 2023, is to make it completely self-deployable so that people can deploy it, run the cloud formation, and have as many accounts as they want to monitor their own projects.
Okay, thank you very much, Dana. My congratulations on your successful launch. I feel that the project is really successful and you proved a few concepts with these hospitals that you mentioned. So thank you very much for participating in our conference. And I think we are ready to move to the next speaker. So
thank you very much.
Thank you, Dana.